Okay, so we are live on YouTube and Google Hangouts with our very first Summer of Symbiosis book club. So I'm really excited that we've got a good group of people here to talk about this particular book, our first book in the series. 1 + 1 = 1, Symbiosis and the Evolution of Complex Life. And I should not be covering the author like I was. It's John Archibald. And so we'll be chatting about this book. And if you are following along on YouTube, feel free to tweet at us. If you have a question, you can use the hashtag G+microjc. Also, if you were trying to join our Hangout and you're confused about it, feel free to tweet at me. I'm at Microwave Side, and I might add that to my little toolbox so that people can see that if they've got a question for me. So I will briefly introduce myself. I'm a new assistant professor at Providence College in Providence, Rhode Island, and I study predatory bacteria and the evolution of bacterial lifestyles, and I've just started doing that, so I'm getting some good help from Mark Martin, who will be showing up later to introduce himself. And I am particularly interested in reading about symbiosis because I'll be teaching a class in the fall specifically on symbiosis for our biology majors here. So I'm really excited to have the opportunity to start thinking about that now so I can think about how I want to shape the class and kind of form the class as we go. So, oh, I see somebody else has joined us. Yay!
Hi!
Hi! So we're just doing introductions, and we are kind of broadcasting to YouTube, so we're hopeful that maybe some other people will be able to hop in once they discover and navigate Hangouts here.
Great.
So why don't we go ahead, and I've got an order along my panel. So Mark is right next to me, so I'll have Mark go next. Would you like to introduce yourself?
Sure. My name is Mark Martin, and I am an elderly associate professor compared to all you young folks at the University of Puget Sound. I have long-standing interests, again, in predatory bacteria, symbioses, and what's especially cool about this particular session is I taught last fall, and this fall again, a freshman writing seminar on symbiosis and predation, symbiosis and parasitism, excuse me. And so this book is a wonderful resource for thinking about that. So I'm quite looking forward to what everyone has to say.
Oh, that's great. Thanks, Mark. Can we go with Kat next, since you're right next to Mark there?
Okay. Hi, I'm Kat Milligan-Myrie. I'm a postdoc at the University of Oregon in the Institute of Ecology and Evolution and the Institute of Molecular Biology. And I study the bacteria in the gut and how the host genetic background influences the immune response to microbes in the gut using stickleback and zebrafish models. And I will be starting a tenure-track position at University of Alaska Anchorage in the fall. And I'm interested in this because I'm very interested in symbiotic microbes and how they live with the host.
Great, thank you. And you gave us our suggestion for our third book, which I see now is going to be, you're going to have some wonderful perspectives on that for what you're doing for the work. That's awesome. Karen, do you want to go next?
Okay. My name is Karen. I'm a PhD student at Simon Fraser University in Vancouver. And right now I am almost on the verge of finishing, and so I admittedly only got through 50% of the book. That's amid all these crazy experiments and trying to finish my degree. But I work on a bacteria called Francisella tularensis, which is a potential bioterrorism agent. And I look at the proteins that are virulence factors and try to characterize and understand how they work.
There we go. I've unmuted myself. Thanks, Karen. And yeah, you know, as Karen mentioned, this is the summer. It's a relaxed or busy time, depending on what you're doing with your summer. So if you're worried that you haven't read through the book, don't worry at all. This is a very informal discussion, so we won't be, as I said before, we won't be giving quizzes. Johanna, do you want to go next?
Sure. Hi, everybody. I'm Joanna. I just defended my PhD a few weeks ago from the University of Southern California. I've been studying Gorgonian octocorals for the past 5 years, so we've got octocorals off the coast here, and I've been analyzing their microbiomes, and I found what appears to be a novel algal organism in their polyps. It's definitely not Symbiodinium, and part of my committee's comments was to contact John Archibald to ask him about this algae. And so when I saw that this was the paper or the book that we were going to be reading, I jumped on top of it. I was so excited. So I'm really actually great. It's really great to meet you guys. I'm excited to talk about the book today, especially in light of my own weird stuff.
Oh, that's fantastic. I didn't realize that your work was going to be so closely related to some of the things that you're talking about today. That's great.
Yeah. If you see on my Twitter page, there's like the main background to my behind my image, and that is the picture of the coral with the golden polyps, and the golden polyps contain that organism.
Oh, that's cool. Yeah, that's really neat.
Yeah.
Oh, great.
So I'm currently working at the Center for Electron Microscopy and Microanalysis here at USC just for the summer, and I'm running a super resolution microscope and an electron microscope preparation, and I'm actually really interested in the visualization of host-microbe interactions in addition to whatever this algae thing is.
Sure, sure. Oh, that's very cool. Great.
How do we set up a thing on ours so that it has like a little banner? Do you know?
Yeah, if you want to go to the left-hand side on the window,
yep.
If you go down to, there's a little thing that looks a bit, it's red, it looks like a little toolbox, and if you hover over it, it will say Hangout Toolbox.
It'll say Hangout to a box?
If you click
Hangout to a box, yeah.
If you click on that, it pops up a thing that gives you some options, and the little person icon on the left gives you a menu for lower third, and you can fill in your name and your affiliation there.
Okay, great. Thank you.
Which is a good idea, because then people can kind of keep track of who is who.
Yeah.
Unless you want to like make comments unidentified, and then you don't have to do it.
It's the Hangout Toolbox, is that right?
Yes.
Sorry,
Yep, no problem.
Okay.
So the book, which we have, I'll show it again. This is like I'm like plugging this book for me.
Yeah, it's great.
So what I thought I would do is kind of give like a brief overview for people who might be watching this and not particularly familiar with what the book is. Maybe they just stumbled across this. So this book is about how endosymbiosis was extremely important for the evolution of the eukaryotic cell, or as it's kind of referred to by Archibald, the complex cell. And it covers the evolution of the mitochondria and the evolution of chloroplasts. And he treats these together and distinctly because they have some similarities in the nature of their evolution, but they also have some distinct features as well. As I was reading it, I kind of felt like the book splits into kind of two sections. One at the beginning is kind of an intent, maybe intended for general audiences to try to bring people up to speed with, what do we mean when we say symbiosis? What do we mean by prokaryotes versus eukaryotes? And also he's careful to lay out the importance of molecular biology to investigating these questions, because a lot of the evidence boils down to ribosomal RNA comparisons. So he's careful in the beginning to try to make sure that people understand, like, why is this data particularly convincing when we get to it, and we're talking about the evolution of these organelles. And then I think the second half, at least this is my impression, the second half of the book really starts to get into kind of what are some of the open questions that we are still investigating about the evolution of mitochondria and chloroplasts. And for me, I think that was great for me to read because I was somewhat vaguely familiar with some of these ideas and models, but not as much as I would have liked to have been. So it was good to kind of see some of this go into detail in a way that I think was pretty easy to follow. But with that as kind of an introduction, I just thought I would, before we kind of delve into details, just ask, what was people's general impression of the book or the parts that you were able to kind of get to? What did you have any thoughts about it in general?
I found the history of the research fascinating because so many times you're introduced to these topics in books or in textbooks as this was discovered and that's all you need to know. But here you have this interesting interplay between these different researchers who had conflicting ideas about how these organelles got into cells and how they did they spontaneously appear or were they actually brought into the cell, and all the background that went into all this. I found the history really interesting, and it made the topic just so much more engaging. So I really appreciated that, and I hadn't even heard of a lot of these researchers because I don't study the eukaryotic cell this deeply. I was a parasitologist in my previous life, but it was really interesting to hear about the background to all these.
Yeah,
stories.
I absolutely agree with you. I think, and I'd made a couple notes to myself that, you know, he was making a conscious effort to tell these stories through featuring different researchers. And he had just enough of the detail about what they were like as people to make it interesting, but not kind of, it didn't overtake the idea of, you know, we're using this as a way to kind of keep with our narrative about what the science is.
Yeah.
I really liked that. And I agree, I have to admit, too, there were names that I thought I did not know who that was, and I'm glad that now I do.
Yeah.
I've been interested for a very long time in symbiosis, and I actually had a couple of interactions with Lynn Margulis back in,
Oh.
and that's a funny story I can tell later if anybody's interested. But I was really thoughtful about how this book has been presented and pitched, because you can do it too simply or you can go too deep in the weeds.
Yeah.
And I thought it was interesting how he split the difference, and he did it sectionally, so that if you get to a place that is too simplistic, you can skip over it if you want more detail, and vice versa. There's a lot of what the gosh wow statistics are in there. For example, and I love this, I made a note to myself, cranking out our own weight in ATP every 24 hours.
Yeah.
And I find with freshmen, that kind of stuff really hooks them. And that's a very, very good thing. I can turn around and say the use of prokaryote will upset, you know, Norm Pace and all the rest. We call it the P word. I like it, but I'm not supposed to say it anymore. I also, it meant a lot to me that he went over stories that I think are really important. I'm sure I'm mispronouncing the gentleman's name, but the Korean scientist Tennessee last name looks like it's pronounced Jeon, with the X bacteria. Very important information. I've followed it for many years, and a wonderful entry to it for anyone who wonders how long it takes for an association to happen.
Yeah.
At the same time, it's possible to nitpick here and there if you'd like, but in all, I think this is a fabulous book.
No, I totally agree. I also and I also agree with Mark in the sense that these, and Kat, that these stories need to be told when you're teaching undergraduates, because I just recently gave a lecture on archaea, and they're interested in the stories. They would, like, we're interested in the stories of discovery, and so if you tell them that in the stories of discovery, it just makes them so much more interested in what's going on. Oh, these people who are scientists, they're real people. They didn't just, you know, they're somewhere up there that we can't reach, but they're real people who are curious and interested, and I think that that's really important to teach them and get across.
And I think it's vital to remind them it's a human profession and has, as we've seen in the news, human frailties involved.
Yeah. I think it was a really important book, not just for teaching other people, but as somebody who has been enveloped in peer-reviewed publications for the past forever, really. And I mean, for my PhD, I haven't been able to step out that much and look at the greater context of things. For me, this book was a really great way, right, to bring those stories, to bring that history, to prevent the reinvention of the wheel, and to also answer the question of why is this what we believe today? Why is this the dogma of what we realize today, and what we all kind of recognize? And it kind of aids me so that I don't have to go back and read all those papers from the 70s, 80s, and 90s. He put it together so beautifully in so many instances of, you know, why things, where we are in our research today and why we're here. And I really appreciated that about this book. And we don't have a textbook like this for endosymbiosis. It's a small chapter in our general biology textbooks, and there's a lot more than what you read in the general biology textbooks. So I appreciated it big time.
Yeah, I absolutely agree. I think this is a really, I'm pretty sure I'm going to use, if not the whole book, definitely parts of it for the class that I'll be teaching. Because I think it's featuring endosymbiosis is a no-brainer in a symbiosis class, and this covers a lot, I think, at a level that the undergraduates will appreciate. So that'll be a great resource. Dee, I'm already doing class prep. Good job.
Well, I mean, if you think about it, too, I read Acquiring Genomes that Lynn Margulis and her son wrote together, and it's very much a as in the style, and again, I say this with the greatest respect to Margulis and her memory, of almost what I would call, a preachment, a sermon.
Yeah.
It really comes across that way. And what I liked here is here are the important arguments not done in that kind of dogmatic style.
Yeah.
Wonderful references to go to, really fair-minded about some stuff that I know could have been unfairly presented. So like I said, really good introduction.
Was anybody else kind of going rocket girl with Lynn Margulis, the whole story that he provided for her? I was so excited about it, especially in the past week with the Tim Hunt news. But I appreciated also their explanation of her and some of the other scientists, the fact that he points out very strongly that they are coming from different stages or from different perspectives to approach one question. And the thing that I really identified with her was, not just asking the question or trying to answer the question from many different perspectives and not just settling in one thing. And I've found that in my own, in myself as a scientist, and I didn't think that that was right. I thought that meant that I was fickle. And it turns out that maybe that's not just the case. That's just one way to do it, and it's not the wrong way to do it. So that was great. I really liked his discussion of her and her work.
Yeah, I agree, and I think that one of the features, certainly in the beginning of the book, when he's talking about kind of historically how have we arrived at where we are now, with understanding how endosymbiosis was a feature of mitochondria and chloroplast evolution. He sets the stage in a couple of ways for people to understand that this is how scientific knowledge builds on itself. There was a description that he had of a meeting, and I'm not going to remember the name of the meeting, but it was a meeting for, I think it was maybe in the 70s.
At Monterey.
Yes, thank you. And there was a discussion, somebody, as he recounted it, someone said, well, why don't we put it to a vote? And Lynn Margulis was like, that's not how you do science. And just the entire tale of that meeting in how people, you have people who kind of occupy poles when it comes to when you're thinking about hypotheses, and then you have people who kind of try to see where the middle is, and then you have, and it all gets kind of worked out in ways that are civil and generous and sometimes not, but that's how you arrive at what gets, as Joanna said, what gets reported in the textbook is like 2 lines of saying like mitochondria have alpha proteobacteria ancestors.
Yeah.
And where it came from are these meetings where people were like, oh, it's this and it's that, and what does the data say, and is that the right data, and you didn't do it right, and this, we should do it again with this.
But that's what we're doing as scientists, right? As scientists, we're trying to define the gray area. We're trying to describe the gray area. And without those tools, how is it going to work to find the gray area? It's great. I think it's fantastic. Yeah.
One of the things, and it comes back to iconoclastic or visionary scientists, for lack of a term, they, talking about Peter Mitchell, referring to him as eccentric, and that is one word for it. But what was good there is the idea of how he continually worked on it and kept pushing on it, and that is how Lynn Margulis lived. And I love some of the comments that were made. That, when she says something that was unlikely or odd, you can kind of discount that, but you should listen anyway, because when she's right, she's very right.
Yeah.
And my favorite Lynnism is the one where they said, someone asked her, how do you feel to be controversial? And she goes, I don't call it controversial, I call it being right.
Yeah.
Yeah. I made a note to myself of the quote, I think it was John Maynard Smith, where the quote was, she was crashingly right.
Yes.
That's a great, I'm just going to start using that as an adjective. That is
crashingly right and crashingly right. Yes, exactly.
I just wanted to make a comment also. I completely agree with you, Joanna, in the sense that coming from different perspectives is so important. And because, right now in the way that we do science, like sometimes I feel that I'm being pigeonholed into this one area where I have to approach things from this particular perspective, but seeing how these scientists would draw from all different areas of their expertise and everywhere around them just really inspired me to think that, okay, I don't have to be in this square. I can expand and think about these things and put them together and create a better hypothesis that way.
Yeah.
Yeah, so I think one of the things that I don't want to skip over necessarily the beginning chapters, but I do think they we've talked a little bit about how he set up kind of the historical context for the issue at hand of exploring endosymbiosis and the evolution of eukaryotic cells. And I do think that for people who are looking for a resource on the history of this is a great resource. It's really accessible. I think it covers names that if you know something about this, you probably recognize from your biology textbook, and it covers people who have probably been ignored. So I think it's valuable to read those chapters. But I thought maybe we could talk a little bit about some of the later chapters, perhaps, where we start getting into kind of discussions of where are we right now. And so there's questions about, and I'm kind of skipping ahead, so if I've skipped something that somebody really wanted to talk about, please feel free to bring it up. But kind of in Chapter 6 and Chapter 7, it talks a little bit about where are we right now with understanding how this event might have happened. Because the evidence suggests that the acquisition of the alpha proteobacterial proto-mitochondrion was a singular event. So the mitochondria involved as a singular event, and that the primary chloroplasts evolved as a singular event. So he makes a contrast between primary chloroplasts and then this idea of secondary endosymbiosis, whereby a eukaryotic cell will ingest a chloroplast-bearing eukaryotic cell and kind of use those for photosynthesis. For the primary endosymbiosis events, those chapters kind of start to talk about kind of where are we right now with understanding how this might have happened. And I think it struck me for the mitochondrion, at least, that the big open question is, which came first? Was it an already eukaryotic kind of cell that got a mitochondrion because you need the cytoskeleton for phagocytosis? Or was the mitochondrion kind of the precipitating event that led to the evolution of a eukaryotic cell? And I just thought I would ask if anybody's got kind of comments or thoughts on that, because I think this is probably the focus of a number of labs' current research, I would say.
That's a good point, because he doesn't discuss too much the production of the nucleus, like the nucleus membrane. And he does a little bit. He just kind of says it kind of off from the outer membrane, but he doesn't go into that. And so I guess, of course, the gray area is where do you start defining a eukaryotic organism, which is what we discussed at the end, right? Is it membrane bound? Really, what else? What are we doing there? And so, is it the presence of the mitochondria that makes it a eukaryote, or is it the beginning of the membrane bound organelles, which presumably would start with the nucleus? But I don't know.
Yeah, I think that this topic could probably be its own much more detailed book, because I don't know, the nucleus as it is now is a complicated structure. Did it start out that way? Probably not. So what did you have a proto-nucleus that was kind of on its way towards becoming what we would recognize now as a eukaryotic cell nucleus?
Well, certainly when you look at some of the work done with planktomycete, where it looks as if there's something like it. I know there's an argument about the topology of it, but I remember a time when I was told there were no types of organelles at all in bacteria.
Yeah, you can still see that places too, which drives me crazy.
And again, without being critical to the author, I mean, the only place I've seen Archibacteria used in recent years has been his book. Right? I mean, that's a really, because they are very different than bacteria, is the whole thing to come back to. He says not so much. I don't really agree. But that's his lookout, and that's fine. I mean, who am I? But I also thought it was interesting when you were talking about this earlier, Laura, about the idea of endocytosis. I mean, bacteria do have a cytoskeleton. They have the equivalent of intermediate filaments, they have the equivalent of microtubules, they have the equivalent of microfilaments. And I would not at all be surprised to see that there are some microbes that are able to actually endocytose. That whole business of how one bacterium gets inside the other is a fascinating one. And you and I know, Laura, that there are ways that can occur.
I was just going to ask you about that because there's, just to take a really brief aside that's going to kind of potentially be selfishly focused on Mark and I. Sorry. Because both of us are working with predatory bacteria. So these are bacteria that eat other bacteria. And the most well-studied one, Delivibrio bacteriovorus, will actually insert itself into the periplasm of a gram-negative cell. And I think there is a recognized predator that has not been studied terrifically well that actually goes into the cytoplasm, isn't there?
That is Ricardo Herrera's work, who I saw at ASM, Daptobacter.
That's okay.
And it's never been cultivated. Boy, I wish someone would just spend some time on that.
Yeah, because I mean there was Eduardo Jurkovic, if I'm saying that correctly, and a colleague whose name escapes me right now, wrote a kind of a paper positing that perhaps it was not the proto-eukaryotic cell that ingested the alpha proteobacterial cell. Maybe it was an alpha proteobacterial predator that made its way into the proto-eukaryotic cell, and so it's the reverse of the way we're thinking about it.
Can you elaborate on that predatory bacteria? It would go into the cell and then explode the cell, or eat the contents inside out, or?
That's what they typically do. So the way they complete their life cycle is they will enter a prey cell, and in the case of Delivibrio, which occupies the periplasm, it actually ends up kind of sealing the cell behind it, and then it proceeds to digest the cytoplasm of the cell that it has infected. It elongates as a filament, and then it septates into progeny cells, bursts the host cell, and releases the progeny.
Wow.
It brings tears to my eyes.
It sounds like a virus.
Yeah, it's a lot like a virus, except it's a bacterial cell that the little progeny have flagella and they swim off and go to attack other cells.
Wow.
And there is a alpha proteobacterial predator that has been described, and that was kind of what prompted these researchers who work on this to propose that, well, maybe we're thinking about this the wrong way. Maybe we're thinking about a cell ingesting another cell and failing to digest it, but what really happened is that the cell went into a cell and then failed to digest it, which is an interesting theory.
That would then support the two membranes that we see actually being from the bacteria, and not potentially from the endosymbiotic. Yeah.
So one of the things that's a big deal to me, and I call it the fault of centrisms. So many people are eukaryocentric. Each freshman bio, you know about it. Then we're oxycentric on top of that. We're only focused on those. And even people like me, old school microbial geneticist like me, gets a little coli-centric. And the simple fact of the matter is we're not looking outside the box. And I mean, I know Abigail Salyers has shown that certain bacteroides are able to grab on to strands of cellulose and digest them. I call that a proto-coated pit, is what I'm thinking of with that. So this might bring us into further ideas of how endocytosis could work among bacterial cells. And the reason that's important is it really ties into how quickly these things can happen. Certainly, you can make the comment that alpha proteobacters appear to be preset for endosymbiotic associations. But is that really true? Lots of archaea can do the same thing. So we need to be looking outside the box. But of course, that's what a lot of people can't do for various reasons.
So I just have a quick question. Has anyone actually observed the integration of a predatory bacteria into its host, where it didn't end up being so it didn't end up being consuming the host, but integrated into the host? Has that ever been seen?
No.
No,
okay.
Not that I'm aware. So you mean similar to the X bacteria that they talked about briefly about being in no, not that I'm aware of.
Okay.
Yeah.
I would love to see that. I will say that there is a type of Delivibrio that makes, I'm going to call them spores, but not really. And they will do that when they're inside prey cells. That's the closest you've got to it. But the idea, and I know that Laura's thought of it because everyone who's interested in Delivibrio does, the idea of something Delivibrio does that the prey cell needs, and that rare event where they're able to have a productive infection, then they'll be able to grow. very similar in a way, if you think about it, to what Gian did with his X bacteria. And most of us have tried it at one time or another without success.
So when you have...
Embryo from? Oh, I'm sorry, Kat.
I was just going to ask, so when you have tried it, have you tried starting out with bacteria infecting another bacteria, or have you tried the eukaryote being infected? I know you're studying a bacteria that eats other bacteria from the inside out, but have you are you focusing on a bacteria incorporating into another bacteria, or a bacteria incorporating into a eukaryote? And that gets back to the question of which came first, the eukaryote ingesting or gaining organelles by taking on symbiotes, or bacteria, one bacteria eating another bacteria and becoming a eukaryote. So in those studies, which one?
Yeah,
What
so
have people tried?
primarily it's for the predatory bacteria. It's primarily bacteria attacking other bacteria.
Right.
I don't know, Mark. Do you know if anybody's tried it with like fungus or something like that?
No, but I think Dave Bolchers would be the man to talk about that.
Right.
Yeah, true.
Yeah.
And he would not be shy.
And he'll be tickled that we mentioned him.
What environment are the Delavibrio from?
You can find them. My understanding is that you can find them pretty much anywhere you look for them.
Okay.
So there are isolated from soil, freshwater, marine. I think there's been some, I don't know if they've isolated them, but people have found sequence signatures from, in like metagenome studies of human feces.
Okay.
Gently put, Laura, you can also find them in very low numbers in the GI tract of certain researchers who remain nameless.
Okay.
That's what I get for mouth pipetting.
Oh, come on now. Don't, yeah.
There's no visualization of them right now. We've not seen them do in their predatory action.
Yeah, and you have to be careful about that business with the 16S, and I really mean this, because I will have friends that will say to me, "Hey, I found Delavibrio in this unusual environment," and I'll say, "Great, you get some platforming isolates." Well, no. I said, "So you have great EM of something?" No.
Yeah.
I just got the 16S, and I'm real nervous about that.
Right. Or just fluorescence in situ. You know.
Yeah, you can see them. You can see them with. Right now, we're trying to get up and running our own like phase contrast scope, because that's kind of the easiest way to see them.
Yep.
But you can get some. People have taken some really beautiful microscopy images that show the attachment and also show the replication of progeny inside a cell.
Why is this? Why wasn't this discussed in the book?
I wondered that when I got there. I was like, oh, I wonder if he's going to mention that. Oh, no, he didn't. Okay. I'm not sure. I think it's a. I don't think it's a widely circulated hypothesis. I think the folks that proposed it made an interesting argument, but I don't know if it really caught on.
Okay.
I know lots and lots of people, Laura, who tell me that it's not possible for prokaryotes to do endocytosis, and I'll say, well, how do you know that exactly? And they get kind of irritable with me.
Mm.
I just think it's coicentricity. You got to look around. You know, look at these L-form style bacteria that don't really have a standard cell wall like we're used to.
Yeah.
They're really variable. They seem to put out what look like pseudopods. So how could that? You know, that would be a good place to start looking, right?
Mm-hmm. Well, and to your point about you know, looking kind of outside the box, I guess, or outside the lab, I guess. I was really happy to read about all, because I was vaguely aware of this, but I felt like it was something I didn't know enough about. Were the variations of so I think for what I can conclude from the book is that calling things amitochondrial might not be particularly accurate, because there's a section in the book that discusses, okay, well, some of the debate was there must be this early branching lineage that doesn't have mitochondria, so that can give us some kind of evidence of which came first, mitochondria or eukaryotic cells. And I think he does a good job in the book of laying out how people went about investigating, well, are they truly missing mitochondria? Can we support the argument that they never had them? And the conclusion to that is no. They have evidence of greatly reduced mitochondria that no longer serve the same purpose that we see with mitochondria in eukaryotic cells. And I thought that was a really interesting section, because that was something that I had kind of knew about, but I wasn't completely informed about where we were right now with understanding those different lineages that have variations on the theme of a mitochondrial organelle.
Yeah, I found that section interesting too, because it pointed out that there are differences in the functions, and that, you know, we tend to think of we have that box for the mitochondria. It produces ATP, and that's its entire function, and it has, you know, it has a certain structure. And that section really highlighted the fact that you can have all these different forms. And I hadn't thought about that before. I didn't even realize that before. So I thought that was really interesting.
Yeah, yeah. And that was yeah, that was a good thing. That was a good thing to read about. I thought that'll be a helpful part for kind of starting to answer that question about origin.
Yeah.
Oh, did we have somebody else who's joined us? Maybe. Sorry.
Yeah.
Looks like somebody else.
Yeah.
Hi, Kevin.
He's getting there. The instant he gets online, we're all going to be like, hey! Okay. So,
I wasn't sure. The thing that wasn't entirely clear to me while I was reading was how exactly the genes from the nucleus of the mitochondria or the chloroplast might end up in the nucleus of the host. What is the actual function that's happening? I'm completely familiar with it, but I've never actually considered the channel, well, not the channel, but the process that happens.
Yeah, I made a note on that too, because it's that endosymbiotic gene transfer. So the transfer of genes from either the mitochondria to the nucleus, the host nucleus, or the chloroplast to the host nucleus.
And I understand he had said, you know, when it gets into the nucleus, then there's the splicing that happens that allows it to get kind of incorporated. I understood that, but what is the physical carrying or transfer from the mitochondrion or the chloroplast to the nucleus?
Oh, that's a great question. Do you mean, like, is it floating freely, or is it somehow chaperoned by something?
Floating freely, presumably, you know, it's getting chomped down by lysozymes, etc., or whatever. But yeah, what is holding on to it and carrying it and allowing it not to get broken down and, like, recycled into the cell and actually getting brought into the nucleus, into the nuclear envelope?
I've read of people who've made claims that if you follow the transit of mitochondria and chloroplasts inside the cytoplasm, they nestle up to the nucleus and then they move away, like there's a pathway to that. Mm-hmm. Now, I know that at least now there are gene products made by the nucleus and gene products by the mitochondria necessary for the mitochondria to work. Right. They have to get there.
Right.
But I would say that sometimes we forget that the cytoplasm is an environment.
Right.
And if you look back to Seth Bordenstein's work with some of the phages of Wolbachia, here you have bacteriophages going from endosymbiotic cell to endosymbiotic cell in the cytoplasm.
Right.
So to me, this is just an unusual environment in which these things could happen. I would have to go digging through literature, but I'm almost certain that there's like a pathway for many of these things to happen. But this takes you back to the chicken and egg argument. How did that originally take place? Might have been accidental, and then there was an advantage to that winnowing that took place.
Well, on that note, that reminds me of, you know, in the beginning chapters where he discusses, you know, being inside of a cell and how it's so compact, and how he has like one sentence that I don't recall where it is in here that he describes it so well, where he makes it sound like a crazy traffic mess in there, and the fact that anything gets done, you know, things have to kind of just run into each other by chance is like a fantastically fortunate thing for all of us. And so I agree, but I guess my thought is if things are so busy and there's a lot of complexity, and yes, the cytoplasm is its own environment, I would think that the chances would be greater that something foreign floating through would get broken down way before. But if you're saying that the mitochondria is actually coming in contact with the nucleus for some short time, well, then that completely helps to decrease the distance of travel.
Oh yeah, and I mean, if you go back, it's not exactly on, but it's related. If you look at Epulopiscium, and there's some wonderful stuff from Small Things Considered. If you don't have time for the papers, there's wonderful discussions of how many nucleoids are present at the periphery of this giant bacterial cell. and that the DNA itself has the viscosity of molasses.
Yeah.
You know, it's a very strange environment, and that's you're hitting my enthusiasm button on this. I have to tell you, newly minted Dr. Holm. But I mean, this is a big deal that we don't think about, and it's important, obviously, if you study this, to think about pathogenesis, if for no other reason.
Yeah.
Because a lot of pathogens live inside cells.
Right.
So anyway, there I go. I'll call it.
I would highly suggest, if any of you have the time and you want to appreciate it more, I just got done with a course on cryopreservation for electron microscopy, and so of course we looked at a lot of EM micrographs, and that just shows you how compact and how crazy things are in there inside of a single cell. Just to visually see it is and then think about it in motion is what really blows my mind.
Amazing.
Yeah.
And I think too, talking about actually the physical moving it across is one thing. And one of the things that I noted when they first started talking about endosymbiont gene transfer, there's a section where, I think for lack of a better term, it ends up discussing like the population dynamics of how this would happen. Mm-hmm. So there are some cells that, for instance, if you use chloroplasts as an example, there are some cells that have multiple chloroplasts. There are some species, eukaryotic species, that have one chloroplast. And so there was a discussion about, you kind of end up potentially at a certain time with a gene, with a kind of a, I don't know, you wouldn't really call that a marrow. In the bacterial sense, you would call that a marrow diploid, but I guess that doesn't work here. So you end up basically with a gene duplication, where one of your chloroplasts has its DNA digested,
right,
and that gene moves into the host nucleus, but your other chloroplasts still have that gene. So for a time, you've kind of got that same gene in two subcellular locations. And I thought that was a really, just thinking about that, you're like, oh, that's really interesting. So you're kind of doing population dynamics on a single cell scale, where you're looking at mutations accumulated and selective pressures for the gene in the nucleus and for the gene that remains in the chloroplast. So that's kind of cool.
Does that mean that it's being transcribed in both places at the same time too? So then that would lead you to a question about the expression, and does the expression increase by 2, so to speak, you know, on a very simplified level, or is it just one at a time, or something? I don't know. Yeah.
Yeah, I thought it was neat to think about it that way, because one of the things that you do, I think he says this a few times, that what you're talking about is such a singular event. All these little kind of chance and necessity things that happened, it's really interesting to think, you know, what exactly was the mechanism by which these organelles. It's somewhat mind-blowing, really.
Laura, I just sent you a link, Laura, and I think you should read the title of it out loud.
Oh, all right. Here we go.
I think Johanna will like this.
Go ahead.
Oh, I'm waiting for it to load. Just 1 second. I'm slow for some reason. Frequent somatic transfer of mitochondrial DNA into the nuclear genome of human cancer cells.
Yeah.
I would read the author list, but it would take me until about 3:30, so I won't. It's a frequent somatic transfer of mitochondrial DNA into the nuclear genome of human cancer cells. So
I tend to think of everything just, maybe I'm becoming more Ghoulian. I think that everything's just kind of moving around. I'm not ready to think that flagella were originally spirochetes, so I'm not there.
Yeah.
He also used to think that nuclei were symbionts too. Sorry. I'm not ready.
That nuclei were what? I'm sorry.
I couldn't hear that.
Lynn Margulis thought that nuclei were also symbiotic in origin. And that I'm not prepared for either. Although, when you think about the red algae that steal nuclei, that makes you think about that, doesn't it? What's the expression? Kleptoplasty? I love that.
They steal the chloroplasts, right? Chloroplasts. Yeah, that was fascinating. So the I had not heard about that, kleptoplastity.
Oh no, I've absolutely been considering it in the case of my corals with their weird things, and it's raised a lot of discussion amongst my colleagues here and myself about so is symbiosis truly at the end of the day mutualistic, or is it actually an enslavement? And the idea of kleptoplasty definitely puts forth that idea that, you know, the enslavement, where something's obviously being ingested and just part of it's being held on to. And then it raises the idea of the foraminiferae, who can also, they actually capture and hold on to symbionts, and that's actually a form of enslavement. And it's just, it opens a whole fascinating discussion, which I actually was happy. He talked about lots of examples, but not until the end, to the very end of the book, because it's an overwhelming discipline of all those types of interactions and the examples of them. And so I much preferred the way he did the book rather than just making chapters about all the different symbioses that are out there and how they may relate to endosymbiosis.
There's this philosophical view of symbiosis as being kind of like all bringing together, but I view it as a rough kind of wooing that's taking place over evolutionary time. I mean, the idea of enslavement is much in my head. So.
Yeah.
So, Joanna, do you want to tell us real briefly about the system and more? Since it sounds like this is very applicable to some of the things that are being discussed. Can you tell us a little more about your system?
Yeah, it hasn't been published yet. I'm working on it.
Okay, well, only tell us as much as you can.
No, I will. But the reason it's not published yet is because I'm still trying to identify the organism, the algae. It would be a much better paper if I can actually tell you, taxonomically speaking, what it is, and phylogenetically what it is. And but it's these corals that are out here, and my original question was, why are some of the polyps golden on some colonies and on the other ones they're white? And I'm a diver, so I would go down to caves really deep, and I would see that only the white polyp guys were down there. And so I thought, well, maybe there's something, you know, photosynthetic happening here, which wouldn't be surprising because Symbiodinium is a very common symbiont in corals all around the world. And so I started doing microscopy because, as I've found, that is my thing. And the cells look nothing like Symbiodinium. They are amorphous. They don't have a consistent shape to them. They look kind of like colored or pigmented jelly in the midst of the coral milieu. And they have different pigments than Symbiodinium. Symbiodinium are dinoflagellates, and they have certain pigments associated with them, and these guys don't have those pigments in them at all. They do have a nucleus, so they are eukaryotic. I've done DAPI staining just to show that. And I've tried many, many times to genetically determine what they are, but unfortunately have either gotten no amplification when I've chosen primers for specific, you know, either photosynthetic genes or just, you know, taxonomic genes, or I get coral amplification, so the coral contamination of the DNA. They're very interesting, though. So I've measured photosynthesis happening at the surface of the coral, of the polyps, and it's relative to the density of the cells found in the polyp. So something's happening in there, and I've been to quite a few conferences in my time here, and the topic of all of my talks has been, you know, about the bacteria or the microbiome, and then at the last 2 slides I put a plug in and I show all the images I have, and I'm like, if anybody's seen this, like the back of a milk carton, if anybody's seen this guy, let me know. And I've yet to find anybody who knows what it is. So my last really interesting piece of information has come from my electron microscopy images, which I have to do more of because the resolution's not fantastic on these. But it looks like their chloroplast has only 2 membranes around it, which is more suggestive of primary symbiosis rather than, you know, the secondary or tertiary, which we see in dinoflagellates. So I'm currently trying to better preserve the samples to get an idea, to get a better look at the thylakoid membranes. But the structure of the cell and the structure, the way the chloroplasts look in the cell is very different than anything we've seen, meaning in a lot of dinoflagellates, or haptophytes even, or promuciophytes, you see the chloroplast as kind of a peripheral structure along the outside of the cell, internal, but along the periphery. And these have chloroplasts all throughout. So something funky is happening there. Whether it's kleptoplasty, you know, enslavement, some type of actual symbiosis is still to be determined. So I think John Archibald might actually be interested in it, and I was suggested to contact him, which I probably will do here in the next few days.
Oh, that's very cool. Yeah, that's very relevant.
Yeah, it's exciting to me, and it's just opened up this whole world of interest for me. So I really enjoyed reading the book, and I was psyched to see that you were going to have this, just to hear more about what the other experts have to say, because I don't know that I consider myself one of those yet.
But I mean, it sounds like a great system for kind of being able to, there's an outside the box right there.
Yeah, it could be a very cool thing, right? Because it's a coral. It's not microbe on microbe happening here. So who knows? And these octocorals are kind of, they're pretty ancient, and who knows what is happening there with all of them.
Very cool. Thank you for sharing that. I think that's good. That gives it, I mean, that's, as you said, like there's a few things towards the end of the book where he mentions kind of, you could dive really deep into all the interesting ways that symbiosis and the interesting systems that you could ask questions about. So I'm glad we got, your perspective on that system in the context of this. That's great.
Yeah, and with the idea of the membranes, that's the thing that's really interesting to me too, which I'm glad he talks about so much in the book. Or, a little bit at the end, but you have to understand the structure of the cell and where the cells have come from to know more of what you're looking for. Did I get a thumbs up right there? Is that what that was?
That's what that was.
Also, I have to go. I have a meeting at noon, so I have to go. But thank you. This was a great discussion.
Yeah, thanks very much for joining us.
Nice to meet you, Kat.
Nice to meet you, too.
Bye.
Bye.
So I thought we are kind of reaching 3, but I wanted to kind of ask if anybody kind of had any other final thoughts about this. I was going to, if I can single out Karen really quickly. Karen. Because Francisella is an intracellular pathogen, yeah? Mm-hmm. So did anything that from this book and the parts that you kind of were able to, given that you're going to be defending in December, right? So you're.
I think so. Hopefully. Cross your fingers.
Staggering towards your defense. But did you, I mean, from the perspective of someone who's looking at an intracellular pathogen who's kind of has an antagonistic relationship with a eukaryotic cell, I mean, were there parts of this book that you found kind of illuminating for what you're doing?
Well, I have to confess, I was zooming through a lot of the book, and a lot of the things that I found interesting were about the stories. But as when we were talking about it in our discussion, when Mark brought up that one thing about the bacteria infecting other bacteria, I was wondering if it would have been the bacteria itself would have been slightly avirulent. So it wouldn't have been as virulent, and the avirulence allows it to create a symbiotic relationship, or it might be lacking specific genes that would be virulent. And then so now that if we think about it that way, then virulence isn't as straightforward as you think it is, because you tend to think of 1 and 2 as an antagonistic relationship, but it seems more like a dance. Whereas in one aspect it might have been antagonistic, so it infected, but then it wasn't virulent enough to infect and take over the cell, so then the cell kind of attacks back, but then eventually over time, as it was talking about in the book, it forms like this symbiotic relationship. And I thought that was really nice, like almost like a relationship between these two people, where at first they hate each other, and then as they kind of begin to understand each other, hate for each other decreases, and then they have like this very nice teamwork relationship. And so
it's the 100-million-year-old rom-com.
There's a danger here, and the danger is the implication that things go in a particular direction. Evolution doesn't do that, and neither did symbiosis. Mm-hmm. So if you want to call it the forbidden dance, the lambada, that can go different ways depending on what's going on, that's probably more correct. But the idea that things start off being extremely virulent and tend to evolve toward less virulence is really common. Okay. A lot of people think that, and I don't believe it.
Okay.
I think you should have a tattoo that says avirulence. Avirulence is not simple. That should be your tattoo, Karen.
Okay. All right. So, sorry.
Oh, I was going to ask, though, isn't it more appropriate to think of it, though, as time goes by, because it's generations, right? We're never just talking about a single cell, so it's generation upon generation. And so really it's just kind of a shot in the dark. It's a game of craps. And perhaps this time, in one of the cells in this one generation, with this kind of interaction happening, something suddenly got switched, whether it was a gene or a metabolite or something, that guy had one up, just a little bit of a one up over everybody else, and so this is still going. So I actually think the dance thing is a little misleading, especially if you're going to talk to younger people, because it's generations, right?
Right.
I know we understand that, but
No, that's a good way to look at it. It's not speed dating.
yeah.
Okay. I mean, because it's not. That was just the first thought that came to my mind, but definitely, I would agree that it would be more complex. That it's probably not as straightforward as you would just think it to be, like a dance that moves forward, and that each individual interaction, like, you never know what the result is going to be.
Yeah.
So yes, I definitely agree with that.
Yeah, and I think you raise an interesting point about, and this was one of the things that I find, I guess, kind of, because I think we just don't know yet, but it's one of the things that I found kind of unsatisfying about what our current status is of developing models about how the event actually occurred, is it's just so hard to know what was the underlying mechanism. And the way it gets described in this book by necessity and anywhere else is really hand-wavy. It's like, well, it got ingested, and over time.
Microbial jazz hands.
Yeah, you're like everything, kind of yada yada, whatever. I don't know. But you kind of want to know, well, what happened? Like, was it? Could you think about, like, okay, well, so maybe in this host cell, if it is actually digesting DNA, and DNA from the, the protomitochondrion or the protochloroplast is going into the nucleus, is that somehow leading to changing the nuclear genome in a way that makes it more amenable to hosting another organism, because maybe those genes, maybe that DNA is inserting and knocking out things that are important. I don't know, like what's actually happening kind of at a genetic level across these generations that's leading towards, because it's kind of being framed as like the system gets more amenable towards being the system we think it is. You're like, that's really circular and unsatisfying. Like, what actually happens? I think it's just such a tough thing.
Most associations that we study are either ancient or extremely new. We don't have the process, so we need a symbiotic rich landscape.
Yeah.
Right?
Yeah, and their grandkids probably.
Yeah.
Well, you raise a really good point, though. I haven't even thought about that. The fact that when some of the genes come in at the beginning of a symbiosis interaction, those are in the nucleus. Those are going to be passed on to the next generation. So what are the, at the next generation, say if the association isn't vertically transmitted in some way, what are the products from that gene? Are they being transcribed or expressed in any way previous to the association happening again? Or do they produce a product that puts out some kind of attraction to that cell? I don't know. I've never thought about that before. That's a really good point.
And this brings us straight back to Seth Bordenstein with his whole genome, right?
Oh.
Yeah. Yeah.
That's true, yeah.
And the corals, the holobiont.
Yeah, there you go, yeah. So we're a little after 3, so we got started a bit late, but I think even so we're kind of approaching an hour. Did anyone have any kind of final thoughts about the book that you'd like to share before we kind of get back to our... It's funny because this stuff is like, I love reading this because it's big picture stuff, and you kind of forget that your daily grind does eventually, hopefully, lead to something that might be maybe a paragraph.
There's some great quotes in here all the way through. And of course the one that brings the title of the book into it. Do you know that? Yeah, shall I read that one?
Yes, please.
The endosymbiosis acts to bring evolutionarily distinct lineages together in a manner that can lead to the generation of entirely new organisms. 1 + 1 = 1.
Yeah, I wrote that out. I wrote that whole quote out, and it says in capital letters, "Big message."
It's the backwards idea of speciation. It doesn't have to go this way. It can go this way.
True. That's a really good point. Yeah.
I think it goes this way.
You
Is
like
what
that?
it's doing.
Is
You
that
know?
what's happening? Like this.
Phylogenetic twister.
Oh, I like that. I like that. Then there's a game you can have your students make.
Yeah, that's true. That's true.
You said phylogenetic what?
Phylogenetic twister.
Oh, yeah.
Did anybody have any? I mean, I think that's a great closing thought, but I wanted to give people a chance to offer something else if they'd like to.
I love there's a glossary there. I love the references, but I love there being a glossary. I think that's really important to have in a book like this.
Mm-hmm.
And I thought the references were quite useful.
Yeah. So I don't. At the very end, maybe I'll see. Is Kevin following along? Did you want to offer any thoughts about the book before we depart?
Can anybody hear me, first of all?
Yes. Yes.
Okay, so I think what might be clear is that I'm extremely new to this Google Hangout thing, and so that's why I've kind of been progressively turning my monitor closer to the wall, because I'm starting to realize that I might need a more personal setting than in the middle of the lab office to have any sort of discussions. But
yeah, but that's where science happens. So you're just talking about science and environment of science.
I guess so. I also seem to be pretty popular today, so that's why I'm disappearing at random intervals.
Okay.
I just like listening for now. I actually didn't have any time to read the book, but I think the next time that something like this happens, I'll be very gung ho at consuming whatever literature you guys have thought interesting. And so the way that I come at this is from an immune system perspective. I'm interested in how the gut microbiota will influence the immune system, especially with regards to transplant rejection, because there's been a bunch of work that shows that there's a myriad of factors that influence the immune system in very, very kind of opposite ways, it seems. And the overall effect of the gut microbiota seems to be kind of an anti-inflammatory effect, and they've kind of narrowed that down to certain genera of bacteria and certain factors. But how we can use that in a clinically relevant setting has yet to be discovered, and hopefully be able to shed some light on that and figure out maybe even how to cure my own peanut allergy, because it seems that the gut microbiota and allergy are very intimately linked. So that's where I'm coming at this from, but I just like listening to people who know a lot more about things than I do. So.
Well, thank you for joining us, and certainly, so I'll give the schedule here because this is a good segue. Our next book is going to be we'll cover that in July, and it's going to be The Symbiotic Habit by Angela Douglas. And I just ordered mine. It was a bit expensive, so I'm going to research to see if I can find, if you don't have a copy of it at your library, I'll research to see if maybe there's a rental option or something like that. It is, glancing through it, a bit more dense than this book. So if people, generally speaking, people who are participating today or people who might watch this later, I definitely encourage you to join even if you have not made it through the book, or even if you just scanned the book, because we are not, as I said, we're not giving quizzes. It's summer reading, so if you sat on the beach and glanced through it, feel free to join us. And then the third book, which seems like it's directly relevant to what Kevin's interested in, we'll be doing Follow Your Gut, which was suggested by Kat, who had joined us but has had to go. And that's by Rob Knight, and so that'll be a great kind of gut microbiome book to think about kind of on a broad scale of symbiosis. So that's our schedule, and I'll be kind of picking a date and posting for July pretty shortly, so hopefully everybody can see that, and maybe some folks who joined us today can join us again. And I'd just like to thank everybody for being here today.
Thank you.
So thank you for a great discussion, and this will be up on YouTube for people to watch for posterity, I guess. So, thanks very much, everybody.
Thank you.
Bye.